Hello everyone, you are watching Blackboard and welcome to our microbiology playlist. Previously, we have discussed about bacterial structure and easy mnemonics to learn bacterial classification. You can find the links in the description box below. Today, we are going to talk about Gram staining, discovered in 1884 by Christian Gram. So it is an important staining procedure in microbiology that divides the bacteria into two major groups based on their cell wall composition, called gram-positive bacteria and gram-negative bacteria. Let's talk about the procedure consisting of only 5 steps. Smear preparation on the glass slide and fixation. Fixation could be done by 3 methods: air drying, heating on flame, and alcohol fixation. This step helps the smear fix on the slide and allows for better staining and differentiation of cells. Cover the smear with crystal violet for 30 to 60 seconds. It is a dye that stains all the cells purple. Wash with clean water. Cover the smear with Gram's iron solution that acts as a mordant. It enhances the binding of dye to the cells. Wash again with clean water. Decolorize the smear with alcohol acetone solution in 1:1 ratio. Because alcohol alone takes longer time for decolorization, whereas acetone alone has the capability to over-decolorize the smear. This step is performed for a few seconds and washed immediately when the smear is decolorized. In the last step, cover the smear with a counter stain such as safranine or diluted carbylfuchsin for 30 to 60 seconds and wash with clean water. So what actually happens is that decolorizer extracts the purple dye complex from the lipid-rich, thin-walled, gram-negative bacteria to a greater degree than from the lipid-poor, thick-walled, gram-positive bacteria. So gram-negative organisms appear colorless, whereas the gram-positive bacteria remain purple. The red dye, safranine, stains the decolorized gram-negative cells red or pink, and the gram-positive bacteria remain purple. There is a simple mnemonic to learn the order of steps. That is, come in and stain, which depicts the order crystal violet, gram iodine, decolorizer, that is alcohol acetone, and safranine. Examine the smear under microscope at low power lens first to check the distribution of cells, and then under oil immersion lens to view the morphology of cells. Gram-positive bacteria and yeast cells will appear dark purple, whereas gram-negative cells will appear pale to dark red. You have to report the gram stain results as follows: the gram reaction, that is the gram-positive or gram-negative bacteria, the morphology of the cells, whether they are cocci, bacilli, or some other morphology. Then we talk about the arrangement of bacterial cells such as clusters, chains, or diplococci. Then the number of cells is reported, whether they are few, moderate, and many in numbers. It could also be reported as 1+, 2+, 3+, and 4+. Here are some results of the gram staining procedure. Gram-positive cocci in chains 2+. Gram-negative diplococci in moderate numbers. Gram-positive cocci in clusters 2+. Gram-positive pleomorphic rods, that is, they vary in shape and arranged in V-shaped formations 3+. Gram-positive rods with square-shaped ends 2+. If the smear is prepared directly from the specimen instead of the agar plate, report the following things: number of white blood cells and red blood cells per high power field, number of epithelial cells per low power field, the morphology of bacteria, and comment on the morphology of fungi as well. Budding yeast or septate or non-septate hyphae may be present in the smear. However, there are some bacteria that cannot be seen in the gram stain, such as mycobacteria, including Mycobacterium tuberculosis, because there is too much lipid in the cell wall so dye cannot penetrate. Treponema pallidum, because the cell wall is too thin to see. Mycoplasma pneumoniae, as there is no cell wall and is very small. Legionella pneumophila, as there is poor uptake of red counter stain. Chlamydia and Rickettsia, as they are intracellular organisms and are very small in size. There are some alternate stains for these bacteria that we are going to discuss in the upcoming videos. The sensitivity of the gram stain procedure is low, as it requires 100,000 bacteria per ml to see one bacterium per microscopic field using the oil immersion lens. So blood cultures are recommended to be incubated overnight to allow the bacteria to multiply. So this was all about today's topic. Now if you have any questions, you can ask in the comment section below. And if you liked the video, subscribe to the channel to stay connected to more medically related content. Thank you.